Hello friends, I am Suchi. Welcome to my YouTube channel. In this video, we will discuss about central dogma. First, we will know what is central dogma. Central dogma is a pathway in which genetical information stored in DNA that will pass on through RNA and express in the form of protein. So, whatever genetic information stored in this DNA that will pass on to RNA or it will copied on RNA, then that RNA will help to synthesize this protein. As you know, most of the living organisms having DNA as their genetical material, and this DNA carry genetic information from generation to generation. Whatever the characteristic one person have or one living organism have, that will pass on to its offspring through this DNA. And most of the DNA having two polynucleotide chain. Suppose this is one DNA, it's having two polynucleotide chain and both run anti-parallel to each other. Suppose in one strand or in one polynucleotide chain, one end having 3 prime, it will face towards 5 prime of other strand. Similarly, one strand having 5 prime, it will face towards 3 prime of other strand, and both the strand or both the polynucleotide chain bind with each other by hydrogen bond. Suppose in one strand you will find nitrogenous bases like adenine, guanine, cytosine, thymine, adenine, guanine. Then in opposite strand you will find their complementary nitrogenous bases. So here A will bind with T, which is thymine, by 2 hydrogen bonds. Similarly, guanine will bind with cytosine by 3 hydrogen bond. So here you will find adenine, thymine, cytosine. So in this way, nitrogenous bases are arranged in DNA. It means nitrogenous bases present in this DNA are carrying genetic information and passing them from one generation to next generation. But here DNA can't directly participate to synthesize this protein. It needs help from RNA. Through RNA only it can synthesize protein. When protein will get synthesized and it will express, through that expression we can guess the presence of codon in DNA. Here information passing from DNA to protein through RNA. So to complete this flow of information, 3 different processes are involved. First one is replication. Second one is transcription. Third one is translation. Protein synthesis completed in 2 steps. One is transcription, another one is translation. Then why DNA will go for this replication process in this flow of information? Why it will go for replication? Because for growth and development, cell will always go for cell division. From one cell, 2 cell will produce, from 2 cell, 4 cell will produce by mitosis process. To maintain the continuity and amount of DNA in cell, DNA will go for replication and it will produce 2 identical copies from one DNA molecule during replication process. to distribute equal amount of DNA of same type. For that reason, it will go for replication. So now DNA will go for replication process, and during this process both the strand will get separate and they will act as template to synthesize their complementary strand. So here it will form its complementary strand. This strand will also form its complementary strand. So in this replication process, two identical copies of DNA molecule will get synthesized from one single DNA molecule. After this replication process, DNA will go for transcription process. During this transcription process, mRNA will get synthesized from DNA molecule. This DNA molecule will go for transcription process. This DNA molecule also will go for transcription process. Suppose for example, this DNA molecule will go for transcription process, and from these two strands, one will act as template to synthesize mRNA in presence of RNA polymerase enzyme. Suppose in these two strands, this strand will act as template. So in this strand, what are the sequence present? So here you will find adenine, guanine, cytosine, thymine, adenine and guanine. So these are the nitrogenous bases you will find, or nucleotide you will find in this strand. When it will act as template in presence of RNA polymerase, it will synthesize its complementary strand. So during this transcription process, whatever sequence you will find in mRNA, that will be the complementary to this template strand. This template strand also known as nonsense strand or non-coding strand. And here which strand is not participating in this transcription process? Suppose in this case, this strand is not participating as template. So this strand will act as coding, because here you will find complementary bases to this strand, which will be equal to this strand or same with this strand. So this will act as coding strand, which is not participating in this transcription process. In RNA, in place of thymine, you will find uracil that will be added by this RNA polymerase enzyme during this transcription process. So here you will find sequence like U, C, G, A, U and C, which is the complementary to this strand or this sequence. Along with mRNA, other RNA also will get synthesized. Those are rRNA and tRNA. After synthesis of RNA, now this mRNA will go for translation process. During this translation process, this mRNA will help to synthesize protein in ribosome. During this translation process, actually what happened? First, this mRNA will get attached at the smaller subunit of ribosome. Suppose this is the smaller subunit, here it will first bind. This mRNA will get bind here at its specific point. Then after binding of this mRNA, one new tRNA will come and bind with triplet codon present on mRNA, because this tRNA will have anticodon, or you can say triplet anticodon, which is complementary to the codon present on mRNA. First it will bind, after binding one new amino acid will get synthesized. After synthesis of one amino acid, next another one tRNA will come and it will bind with next triplet codon present on mRNA. So in this way, new amino acid will get synthesized and they will bind with each other by peptide bond. So here peptide bond will form and they combinedly form a polypeptide chain. And form a new protein. Whatever the genetic information present in DNA, that will pass on to mRNA. From mRNA, protein will get synthesized. So in the form of protein, that genetic information will express. It means here information or genetic information flow is unidirectional. In one direction it will flow, it can't be revert. For that reason, this pathway named as central dogma. It was first proposed by Francis Crick in 1957. Here pathway follows these three steps. First one is replication. DNA first replicate and it will form number of copies. From that DNA, mRNA will get synthesized by transcription process. From mRNA, protein will get synthesized by translation process. So it is the pathway of central dogma. Here you will find nucleotide in DNA arranged in linear manner. Here in polypeptide also amino acid arranged in linear manner, and those amino acid are present as triplet codon in mRNA. It means whatever the sequence you will get in this polypeptide chain in the form of amino acid, similar sequence you will find as nucleotide in this DNA. That is the collinearity property of DNA and polypeptide chain. So here you can face one question. That one is the pathway of central dogma, which is having three process. One is replication, DNA to DNA replication, then DNA to RNA transcription, then mRNA or RNA to protein is translation. Another one question also you can face, that one is who has proposed this central dogma? Answer is Francis Crick in 1957. This is all about central dogma. Hope you understand. Please like, share, comment and subscribe my channel. Thank you.